Okay, so welcome to this next video in the playlist on calcium signaling. So far we've looked at skeletal muscle contraction, and we've also looked at excitation-contraction coupling in cardiac muscle cells. And both of those calcium signaling pathways involved calcium being released from this intracellular store of calcium in the endoplasmic reticulum or sarcoplasmic reticulum, since both of those examples were muscle cells. Okay, now what we're going to look at is another example of calcium signaling involving calcium being released from intracellular stores, i.e. from the endoplasmic reticulum, but this time it happens in loads and loads of different cells. It doesn't just happen in excitable cells such as myocytes or muscle cells. Okay, so basically the topic for this video is the phosphoinositide pathway. So we're going to look at how G-protein coupled receptors can activate a pathway known as the phosphoinositide pathway, and how this can lead to the release of calcium from the endoplasmic reticulum. So the phosphoinositide pathway. Now, we're not going to look at the IP3 receptor in a huge amount of detail in this video. We're going to save that for the next video. So this video will be the pathway that leads to the IP3 receptor, and then we'll study the IP3 receptor in its own right in a separate video. Right, okay, so let's begin then. So let's say we have the cell membrane here, which is the phospholipid bilayer. So here are the two layers of phospholipids denoted by these two lines. And then what we have in here is a G-protein coupled receptor, so a 7-transmembrane receptor, which has 7 transmembrane domains, like so. So this is a G-protein coupled receptor. Okay, often abbreviated to GPCR, is equal to a G-protein coupled receptor. And basically, these G-protein coupled receptors, which activate the phosphoinositide pathway, will be coupled to a very specific type of heterotrimeric G-protein. Okay, so I'm going to keep it nice and general at first. We're going to talk about general G-protein coupled receptors, although good examples of G-protein coupled receptors that are going to activate the phosphoinositide pathway is the muscarinic 1 receptor. M1 is a key example as far as history is concerned, where these pathways came from. M3 is also very important, so muscarinic receptors are very, very important for acetylcholine. Okay, so there are examples of GPCRs that would be coupled to this signaling pathway intracellularly. Okay, so what G-protein does this GPCR interact with? Well, let's draw our heterotrimeric G-protein here and we'll find out. So a heterotrimeric G-protein consists of 3 different subunits: an alpha subunit, a beta subunit, and a gamma subunit. And basically, you can make loads of different types of heterotrimeric G-proteins because there are 16 different alpha subunits, there are 5 different beta subunits, and there are 12 different gamma subunits. So there are loads of possible combinations for heterotrimeric G-proteins which you can make. Now, the name of the G-protein is completely determined by which alpha subunit you use. So if we're looking at the heterotrimeric G-protein GS, then what that means is that you have the alpha subunit is the alpha S subunit, and we don't care what the beta or the gamma subunit are. Now, the heterotrimeric G-proteins that are going to be coupled to our GPCRs and which are going to activate this phosphoinositide pathway are GQ or G11 G-proteins. So there are 2 different G-proteins which can interact with this phosphoinositide pathway. And if you are a GQ G-protein, it means that your alpha subunit is alpha Q. If you are a G11 G-protein, it means that your alpha subunit is the alpha 11 subunit. And we do not care which of the 5 beta subunits you use and which of the 12 gamma subunits you use. If you are a GQ protein, your alpha subunit is alpha Q, and the beta and gamma can be whatever you like. If you're a G11 protein, your alpha subunit is alpha 11, and your beta gamma subunits can be whatever that you like. Okay, so for the phosphoinositide pathway, we are talking about GQ or G11 G proteins, where this alpha subunit is either Q or 11. Right. Okay, so this G protein coupled receptor, when it's in the inactive state, sometimes G protein coupled receptors are actually physically linked to their inactive heterotrimeric G protein. At other times, the heterotrimeric G protein is bound to the inner leaflet of the phospholipid bilayer, i.e. the inner layer of phospholipids in the phospholipid bilayer. And basically, when this GPCR becomes catalytically active, when its ligand binds to it, so I'll draw its ligand binding to it. So here comes the ligand has bound to the GPCR, and when that happens, the GPCR becomes catalytically active, and it's going to catalyze a reaction involving your G protein. Okay, so let's say we have either GQ or G11, which are the important ones as far as phosphoinositide pathways are concerned. So in the case of M1 or M3, they're coupled to GQ generally. But phosphoinositide pathways can be activated by G-alpha Q or alpha 11, basically. Now, when the G protein is in its inactive state, then the alpha subunit has bound to it a GDP molecule, okay? So, in the inactive state, the heterotrimeric G protein is all bound together, the three subunits are bound together, and the alpha subunit has a GDP bound to it. Now, when the ligand binds and the GPCR becomes catalytically active, it's going to catalyze the breaking of this GDP molecule and the attaching of a GTP molecule to that alpha subunit. So you're going to get GTP bound to your alpha subunit. Now, when you bind GTP to the alpha subunit of your heterotrimeric G protein, which has either an alpha subunit Q or 11, so when I write alpha Q/11, I mean it's either alpha Q or it's alpha 11. I don't mean some combination of both. Okay. Anyway, when this GPCR becomes active, it's going to catalyze the reaction which breaks this GDP alpha bond and basically brings in a GTP molecule and replaces that GDP molecule with the GTP molecule. And now once the alpha subunit has GTP bound to it, it no longer wants to associate with the beta and the gamma subunit. So off the beta and the gamma subunit go. And they remain bound together, but the alpha Q/11 is off doing its own thing now. Okay, so what does this alpha Q11 bound to GTP go off and do? Well, it goes and activates an enzyme that is within the membrane of cells. Okay, so let me draw down here the enzyme which it's going to activate. So here's the phospholipid bilayer, and basically in the phospholipid bilayer there will be an enzyme called phospholipase C beta 2. Okay, and that's basically the enzyme that this is going to go and activate, phospholipase C beta. In fact, actually get rid of that too, we'll just have phospholipase C beta. So there are many different types of phospholipase C. Phospholipase C beta is the type activated by alpha Q/11 G proteins bound to GTP. So basically, alpha Q/11 GTP is going to come and activate this phospholipase C beta. So let me just write that name out. Phospholipase C beta. Okay, so once you've activated phospholipase C beta, that is now going to carry out, carry out a reaction in which it breaks down a molecule known as PIP2, which is constitutively found in the cell membrane, into IP3 and DAG, and we'll look in more detail in the next video at what that means.